is done by semi-conservative replication, which means keeping one strand and building a new complementary strand based on the first one. Initially, the enzyme DNA helicase unwinds and separates a portion of the DNA molecule to create a replication fork. Single-stranded binding proteins react with and stabilize the separated single-strand sections. Two new strands are made by pairing bases to the template strand. The enzyme primase lays down an RNA primer which signals DNA polymerase III to start building the replicated strand. DNA polymerase I comes in to replace the primase and fill in the missing nucleotides from where the primase was. The new strand is created in fragments called Okazaki fragments. DNA polymerase I also proofreads the new DNA to make sure that no mistakes were made. DNA ligase then connects the sugar phosphate backbone of the DNA to bond where polymerase I and polymerase III build. In the end, we are left with two new strands of DNA, both with half of the old DNA and half new DNA. Transcription, which is the synthesis of messenger RNA, or mRNA, involves three stages: initiation, elongation, and termination. The RNA polymerase binds upstream to the section of DNA being transcribed. The region it binds to is called the promoter region, which is a string of A's and T's. In elongation, the RNA polymerase starts building the single-stranded mRNA in the 5' to 3' direction. The polymerase transcribes the template strand to create a new strand of RNA. The RNA polymerase will stop coding the RNA when it reaches the terminator sequence. Once it reaches this point, the mRNA detaches from the DNA strand, creating a brand new single-stranded mRNA.
Translation is the process of converting mRNA into amino acids to form proteins. Each amino acid is coded for by a combination of 3 mRNA nucleotides called codons. Translation starts by 2 parts of a ribosome binding to the start codon, AUG. The ribosome then moves along the mRNA reading each codon and signaling transfer RNA, or tRNA, to bring the corresponding anticodon. The tRNA enters the P site and the next tRNA enters the A site. With both molecules there, the tRNA in the P site transfers its amino acid to the tRNA in the A site. Then the molecules shift and a new tRNA enters as the first leaves. This cycle continues until the ribosome reaches a stop codon and the new amino acid chain detaches as a finished protein.